Section 2, mitosis and cytokinesis. These are stage 2 and 3 of the cell cycle. Are you guys aware that your body will produce about 2 trillion cells every single day? Wow, that cell cycle must be working out. Actually, most of these are really just skin cells. Different types of cells will divide differently. If you're a prokaryote, you divide by binary fission. If you're a eukaryotic cell, you divide by the process of mitosis. The main goal of mitosis is to produce 2 identical daughter cells with the same number of chromosomes. If this goal wasn't accomplished, we would have a bunch of cells that are not the same, may be mutated, and could even perform the wrong function. Not a good thing. Don't forget, the cell cycle will not work unless there's a need. And again, those needs are for repair or growth of a body cell. The animation is showing mitosis, which we will break down shortly. This is a diagram interphase and then the 4 steps of mitosis. A hint of how to remember the different steps is to remember PMAT. The first step of mitosis is prophase. As you can see by the cell picture, a few things have happened. The nucleolus has disappeared. The nuclear envelope has dissolved. This allows the chromosomes to condense or become visible since they are usually hidden inside the nuclear structure. So no nucleolus, nuclear membrane is dissolving. You can see the chromosomes. The other important part is the spindle fibers have been completely formed. Here are our spindle fibers. Here is our centrioles, which also have been made. The next step of mitosis is metaphase. As you can see by the cell picture, something also has happened. The sister chromatids have been pulled to the equator of the cell by the spindle fibers and the centrioles. They have aligned all the sister chromatids, aka the chromosome, to the middle of the cell. So here's my chromosomes, my spindle fibers, and my centriole. Metaphase, middle, MN, one way to remember. The third step of mitosis is anaphase. As you can see by the cell picture, something new has also happened. The centromere has been split. The sister chromatids have been pulled apart towards the opposite poles of the cell. So here you just have half of your chromosome, or your one sister chromatid, with your other sister chromatid on the opposite, because it was pulled by the spindle fibers and your centrioles. Kind of think of it as like Spider-Man when he shoots out his web. His wrist would be like the centriole. This is the web, and then it's hooking on to whatever he needs to hook onto, but here it would be your chromatids. The fourth step of mitosis is telophase. Again, you can see by the cell picture that something has occurred. The nucleolus reappears along with the nuclear envelope reforming. Because these are back, the chromosomes cannot be seen and are called or are considered decondensed. The spindle fibers have dissolved and the cytoplasm begins to pinch inward. So here's your nuclear membrane, all back, and then you can see the slight pinching of the cytoplasm. The last stage is cytokinesis, the end of the cell cycle. The cytoplasm completely divides in half, allowing the cell membrane to regrow around each of the cells. This results in 2 identical daughter cells. Please make sure that you are consistent with saying the result is 2 identical daughter cells. In cytokinesis, animal and plant cells are slightly different. In animal cells, we call the cytoplasm splitting at the furrow, which is right here, where a plant must also form a cell plate, which is a cell wall forming. So here's your cell plate because it has to form the cell membrane as well as the cell wall. Prokaryote cells have an entirely different process, which we call binary fission. It is a very fast and a very simple process, since prokaryotes themselves are quite simple. Here it goes. You ready? The DNA is copied, and then the cell divides. That's it. Pretty easy. Here's a little better breakdown of binary fission. The cell is going to get a little bit bigger after the nuclear, the DNA has been copied. Then we're going to go to where the cell wall and the cell membrane are going to begin to split into 2 cells until they completely separate, and then you have 2 cells. A lot of times you will call these clones of each other. Some essential questions for Section 2. List the steps in mitosis. Describe each step of mitosis. What is the final result at the end of cytokinesis? Some vocabulary. Prophase, spindle fibers, metaphase, anaphase, and telophase. Section 3, cell cycle regulation. With all things, some things do not go correctly. In this case, there is normal cell growth seen by the top picture of all identical cells lined up in perfect, neat rows. But then, the cell cycle goes wrong, and we call this cancer. The bottom diagram shows cancer cells among the normal cells. The normal cell cycle is regulated by proteins called cyclins, which bind to enzymes called cyclin-dependent kinases. And if you remember, something ending in ase is an enzyme. These create checkpoints to make sure that the cell and the DNA are copied correctly. With the normal cell cycle, we have checkpoints. The first checkpoint, or the G1 checkpoint, occurs towards the end of G1. Thus, that's why we call it G1 checkpoint. This checkpoint makes sure the cell is healthy and that the current DNA is free of damage and is correct. The G2 checkpoint makes sure the DNA that was just replicated in S phase or synthesis is correct and also free of damage. If it is wrong, it will correct it. It also begins to prepare for the nucleus to divide for mitosis. The last checkpoint is the mitosis checkpoint. This makes sure the nucleus divides and is divided correctly, meaning if the original cell has 3 Golgi bodies, 2 rough ERs, 2 smooth ERs, and 10 chromosomes, the new cell better have the same thing. Sorry about that. This is a cell game that I'd like you to try. Please go to the website and try it out. We will also do this in class. The abnormal cell growth is what we call cancer. Cancer, by definition, is the uncontrolled growth and division of cells. The diagram demonstrates this out of controlness. So you can see normal cells are yellow, and then here these have gone out of control and have created what we call a mass or tumor. Types of cancers. Does cancer only happen in males? Does it only happen in females? Does cancer only happen in certain types of tissues or organs? Does cancer only happen when you get old? Nope. It is not biased. It can happen to anyone, at any age, and anywhere in their body, as you can see by the diagrams. When cancer happens, it means a few things. Checkpoints have failed. It even has the potential to kill, since it crowds the normal cells and does not allow them to do their correct jobs. Thus meaning that your tissue or organ can lose its control of its normal function. This is all caused by mutations that change the DNA. Normally, the body's immune system would destroy cancer cells. If they slip past all the regulators and all the checkpoints, then the cell will continue to divide and make only cancer cells. This forms a mass, or what we call a tumor. There are two types of tumors. Benign is when the mass stays in its original spot, meaning if it is lung cancer, then it only stays in the lungs. This is usually removed by surgery. However, the other type is called a malignant tumor. This means it has spread to other areas of the body. Now, if you had lung cancer, it may now go to the breast or maybe even to the brain. If cancer has spread, we call that metastasis. Carcinogens are substances or agents that are known to cause cancer. For example, smoking may cause lung cancer. UV rays can cause skin cancer. The other one is actually heredity. If you or someone in your family does have cancer, you yourself are twice as likely to maybe also get some type of cancer. One of my favorite words, apoptosis. Apoptosis is programmed cell death. Cells have the ability to program themselves to die because they understand that they are mutated and are bad. This can help to reduce the amount of cancerous growth. Stem cells. In today's world, we have the ability to grow cells to replace ones that have been lost or damaged. These are called stem cells, and there are two methods: embryonic stem cells and adult stem cells. We will go in a little more detail later about these. I just wanted to introduce these terms to you. We will be doing a cancer lab and some essential questions. What is the role of cyclin proteins in controlling the cell cycle? How does cancer relate to the cell cycle? What is the role of apoptosis? What are the two types of stem cells? Vocabulary: cancer, carcinogen, apoptosis, and stem cells.